My name is Kristen Scott. I'm on the board of directors for the NPA. I'm also a research scientist at Moffitt Cancer Center. And I have the honor to introduce our keynote speaker, Dr. Lydia Villa-Komaroff, today. Dr. Villa-Komaroff's career is so illustrious that it would take me the entire hour to do her accomplishments justice. So I'm just going to touch on a few highlights this morning. Her career has spanned from the lab to faculty at prestigious universities, to administration, to industry, to consulting, and nonprofit work. Dr. Villa-Komaroff has been an inspiration and passionate advocate for diversity in the sciences throughout her entire career. She is co-founder for the Society for Advancement of Chicanos and Native Americans in Science, or SACNAS, as most of you are probably aware, and has been an integral part of their 45-year existence, which is absolutely amazing. Dr. Villa-Komaroff has won many awards for both her scientific and advocacy work, and notably, and because it's my current hometown, she was named National Hispanic Scientist of the Year from the Museum of Science and Industry, which is affectionately known as MOSI to my fellow Moffitteers. Interestingly, she was only the third Mexican American to receive a doctorate in the United States, from MIT no less. She studied under the tutelage of Harvey Lodish and Nobel laureate David Baltimore. But it is her postdoc story that I want to leave you with. After finishing her Ph.D., she did postdoctoral work first at Harvard, and when they banned recombinant DNA research, she had to move to Cold Spring Harbor, and then move back to Harvard. She led a team that cloned insulin and figured out how to produce mammalian insulin in bacteria. This is now used by millions of type 1 and type 2 diabetics around the world. Her postdoc work and all the work that she's done throughout her career is such an inspiration and shows what is possible when you put your mind to it. So please join me in welcoming Dr. Lydia Villa-Komaroff.
Good morning. I try to be only half hidden here. I'm short, the podium is tall. In the short time that we're together today, I would like to tell you a little bit about my career path, some of the mistakes that I've made that you can avoid, and then I'd like to talk a little bit about something that has become very important to me, which has helped me to understand why it is that we've been so slow to reach gender equity and representation of underrepresented groups in our various fields. So here we go. I wanted to be a scientist since I was a small child. I'm not sure I understood what a scientist was, except that I think I had this vision of a small, empty, quiet room. I am the eldest of 6 children from a very large family. I have over 100 first cousins. So I was undeterred by things, for example, by being the only girl in a physics class. That just didn't much bother me. And I think that turned into a good mechanism, which is if you don't show that you're bothered when somebody does something inappropriate, it takes away the power of that insult. Well, I left New Mexico to go to the University of Washington as an undergraduate. And while I could not have stated why then, I think I understood that if I did not leave New Mexico at that point, I would never leave New Mexico. And so I went off to the University of Washington, where my father had a brother, and we had visited the World's Fair. While there, as a sophomore, I met Tony Komaroff, who was a medical student. That relationship grew interesting enough that when he went to Washington to join the health services during the Vietnam War, I looked for a place to transfer. I couldn't go to Johns Hopkins because that was all male at the time, and so I enrolled at a small women's college called Goucher College, which was very fortunate in many ways. First of all, I met Dr. Gardner Moment, pictured here. I'm showing you my mentors because I think it's important to realize that through our lives, particularly if we are members of underrepresented groups, our mentors won't necessarily look like us, they won't necessarily have shared our life experiences, but they are nevertheless can be incredibly important to our future. The major thing to look for in a mentor is someone who believes that you can do it, who makes decisions about you based on what you actually do instead of first impressions when they meet you. And you'll learn more about why that's important later. Gardner and other members of the biology department there got me a job working for Loretta Levy at the National Institutes of Health. Now that was very fortunate. I knew that I was going to go to graduate school in Boston because Tony was going to do his residency there. Our relationship had gotten more interesting at that point. And so I applied to every school in Boston almost and gave those papers to Loretta to fill out the recommendation forms, and she said, "Where's MIT?" And I said, "I can't go to MIT." And she said, "Why not?" And I said, "Well, I'm not very good in math. It's not my strong point. I have to work really hard at it, and there's all those guys running around with slide rules," which tells you how old I am. And she said, "You have told me you want to do molecular biology, and if that's the case, then you have to consider MIT, because they have the best molecular biology department in the country and probably in the world." And so reluctantly, I applied. And it's a good thing, because they were the only school in Boston that accepted me. Which is another important lesson. You will not get what you do not ask for. At MIT, I felt like I had fallen into graduate school heaven. It was truly, and still is, a wonderful place to be a graduate student. The graduate students are junior colleagues there. We don't know as much, but we are valued for what we bring to the institution. And while there, at the time, there were no rotations. We went 2 years of classes, and then the faculty came and talked to us, and then we went and talked to the faculty and negotiated a position. Well, before that, my classmates and I sat down, and we kind of divvied up the faculty. We resolved any conflicts we had so that we didn't have any competition when we went to see faculty members. So I went to see David Baltimore, who was already quite renowned, even though he's quite young then. He just turned 80. He had just discovered an enzyme that copies RNA into DNA, and so we all knew he was going to win the Nobel Prize, which he subsequently did. David had a big lab, although he'd only been at MIT a couple of years. And so he told me there was no room in his lab. So I promptly went next door to Harvey Lodish. They were shared an office area, and they shared lab space. And I said to Harvey, we could do a really interesting project. Harvey at the time was interested in cell-free protein synthesis, which was a big deal back then. And so we cooked up a project whereby we would look at the translation of viral RNAs in vitro. And then I went back to David and told him the story, and he said, fine. And so I was the first graduate student at MIT to have 2 advisors who followed and signed my thesis. And I learned, I think, my first lessons in management, other than the ones I had learned as the eldest of 6, in managing David and Harvey as I went through my graduate career. It was a very productive time. I had 5 papers, the most important of which showed that the large RNA in poliovirus had a single initiation point, and so the proteins of the virus had to be done by cleavage and not by translation. At the end of that period of graduate work, Harvey came to me and said, if you go to the Federation meetings in Atlantic City, we'll let you go to the Squaw Valley meeting. I said, yes. In those days, I skied. And so I went, and I learned of this group of people who wanted to discuss why there were so few, in particular Mexican-American and Native Americans in science, because at the time there were fewer than 20 people who were of Mexican lineage or Native American who had PhDs in the hard sciences in the country. The group met, there were about 43 people, and that's because other Hispanics got involved. And at that meeting, we decided that one thing we could do would be to have an annual meeting whereby we would tell students about the benefits of being a scientist and propose it as an alternative that they might consider. Since most Hispanic and Native American students, when they considered college, thought about being a teacher or a doctor or a lawyer, and very rarely considered a science. And so thanks to funding that we got, because J.V. Martinez was at the Department of Energy and David Lopez was at NSF, we got started and began a long journey, which certainly had its ups and downs. The beginning of an organization, as many of you in this room know because you're relatively new, is a rocky one. It's kind of a life-death experience, and sustainability of the organization is always an issue. But we did get started. We designed a logo, and 40 years later, in 2013, you see many of the same group members. There's two things you'll notice right away about the difference in the pictures. For one thing, there are more women in 2013 than there were in 1973. There was one other woman. I have to correct an error that's in the web. I wasn't the third Mexican-American woman to get a Ph.D. in the sciences. It's not clear if I was the fourth or the fifth, but it's out there, so I can't get rid of it. And so we got funding. The institution has become stable, but institutions are living things. Their life is not guaranteed. It requires constant work and attention, and understanding that an organization is not the sum of the people that make it up. It is something different from that. It has a life of its own. And so watching an organization grow and change in response to things is an interesting and important part of what I think I've done. Well, after graduate school, I had to figure out a postdoc, and so I had wanted to do developmental biology all along. So I went to go to see Fotis Kafatos, who was down the street at Harvard, who was studying the development of the eggshell of the silk moth. It's very beautiful because the eggshell is made out of proteins which come on and are laid onto the egg in a very specific and regulated fashion, and it's a time course because they are strung on a little string. You can dissect this time course out of the moth. Unfortunately, we didn't realize that Polyphemus had a very, very large genome. It's as big as the human genome. And so we were trying to use the early methods of recombinant DNA to copy those RNAs from the silk moth and to turn them into something we could use in studies. And we were also trying to clone the chromosomal DNA. And that was before Tom Moniatis had invented phage packaging. In fact, I like to think that the failures that I had helped him come up with that idea because recombinant DNA was banned in Cambridge. There was a great fervor then. These things come and go in science. Things come up. We're in such a time now where scientists are looked upon in askance and doubted, and things can get difficult in the community. So I packed up everything and moved to Cold Spring Harbor for a year with Tom Moniatis, who's shown here in his two incarnations, young and older. And we went off to Cold Spring Harbor, where I spent a year where nothing worked. I mean, it was a really awful, terrible, very bad year. And so after a year, I came back with a total failure with Polyphemus, and Argyros Stratiadis, who was also in Fotis's lab, and he and I were lab mates. We spent a lot of time yelling at each other on the floor, arguing about important things like how big the gel should be, how much triphosphate should be used, and so forth. But networking, like with Argyros, was an important part of what you should do. All the time, when I was at Harvard early in my career there, a graduate student came down and wanted some help with protein synthesis. And I gave it to her, and she went back upstairs, and I didn't think about it for many more years. She'll come back into this story. So Arg introduced me when I came back from Cold Spring Harbor. He said, "Why don't you join us? I'm doing this project with Wally Gilbert, and we need someone who knows how to clone." So I said, okay. And that started off probably the most exciting 6-month period in my career in terms of science. After a year of nothing working, everything worked the first time. This is a rare, rare occurrence. And we enjoyed every minute of it when, we weren't arguing. So I didn't really meet Wally until 3 months into that 6-month period because he was doing a lot of things, and we sort of didn't know that he was also talking about starting a company. It was during this era, and in part based on the work that we did, that Biogen was founded. Wally himself is something of a polymath. When he became a biologist, he was already a tenured professor of physics when he went over, and then, of course, as you know, he won the Nobel Prize shortly after this work was done for his invention of chemical sequencing, which nobody uses anymore. And now that he's 86, he's exploring the world of art and has become something of a renowned photographer and a good friend. So that work opened all kinds of worlds and got me my first job, which was at UMass Medical School. There I made a classic mistake, which you should definitely avoid, which is that I knew that the most important thing that a research scientist in a research-intense institution should do is research. You have to write the papers and get the grants and so forth. But I was on lots of university committees, on lots of curriculum committees, on lots of thesis committees, and there are only 24 hours in a day. So that meant that the work in the lab went more slowly, which meant 6 years later that tenure was not exactly a slam dunk. I did get tenure, and so I decided to go back to Harvard. So I left my tenured position and went back to Harvard, where I had a really good time at Children's Hospital running a soft money lab. I had to bring in most of the money to support me and the people in my laboratory, but it was a really terrific time. I made that move for the wrong reasons. It was the right decision for me, but I made it for the wrong reasons. Good science doesn't depend on where you are. It depends on the effort that you put into the work that you are doing in your own laboratory. And it took me this move and subsequent thinking about things to realize that. So it sort of pays to know why you're doing something. We did a lot of interesting things during that time. For example, we provided the first evidence that beta amyloid is a poison to neural cells. It's not clear yet if it's causative in Alzheimer's. That debate goes on. And there were many other things. To my great delight, now that ResearchGate is available, some of these papers, including the insulin paper, continued, to my astonishment, to be read and cited. Well, after 8 years at Harvard, I began to feel restless again. The funding situation was getting more tense. I figured I needed to focus on one thing. Focus is a good thing. But I talked to people about where I might best use the things that I had learned. And as a result of those talks, one of them was with Alice Huang, who had left her lab to go into administrative work when David Baltimore, her husband, took the presidency at Rockefeller University. As a result of that conversation, we learned, I got a phone call from a headhunter who said there's a job at Northwestern University as Associate Vice President for Research. And my immediate response was, I can't go to Chicago. I'd never been in Chicago except at O'Hare. So she was a wise woman, and she said to me, look, I know you're interested in this kind of a job. You don't have to take this job. But if you're interested in this kind of a job, you have to look at this job. So I talked to Tony, because I knew there was no way he was going to Chicago. He had a professorship at Harvard and was happy as a clam. And so we talked about it. His sister overheard this and said, you know, you guys never travel together. You're always working. You'll probably see each other more if you're in different cities. And Arj gave his inimitable advice, which says, of course, in my opinion, the best marriages are in separate beds, in separate bedrooms, in separate houses, in separate cities, separate countries is going too far. So I went to Northwestern, met Bill Kern, a theoretical chemist who was vice president at the time, a remarkable administrator. He taught me a lot of the intricacies of being an academic administrator, what that meant, what the limits of what we can do were, and what the possibilities could be if you were willing to do that. And so I took the job. Besides, Northwestern beat Notre Dame that year and went to the Rose Bowl. Seemed like a sign. Another key person I met there, other than Henry Beenan, shown in the matching white suit there as we broke ground for a new building, was Don Jacobs, who was dean of the business school. Don called me up one morning, and when a dean calls, I was vice president for research at that point, when a dean calls the vice president, generally he wants something or she wants something. Business school doesn't need anything from the university. They're rich, very rich. So I went to breakfast, because breakfast at the Allen Center is always very good, and it turned out that he wanted me to take the 1 month, 24/7 business school in a box. It was a great gift from him, and it stood me in good stead later. After 7 years, I returned to Boston. Tony and I had been commuting. That was kind of nice to have a little mini honeymoon after every other week, but it was time for us to be in the same house, in the same bed, in the same city for now. So I went back. That graduate student I mentioned a few minutes ago, turned out that was Susan Lindquist. And she had just been recruited to be the director of the Whitehead Institute in Cambridge. And she asked me to come be her vice president, and so I had a job. That lasted 3 years because Susan is an absolutely brilliant scientist, as you may know. She recently passed away from cancer. Really, truly one of the great scientists of our time. And she decided she was better at doing full-time science than being an administrator, and so I began to look around. But in the meantime, I had joined a for-profit board, a company that was its stock was doing well. It was finding enzyme treatments and making them for rare diseases, and it got offered to be bought by Shire Pharmaceuticals, and that split the board. And the chairman of the board and the CEO began to not be able to communicate, and so a new board chair was needed. And we all assumed Wally Gilbert would be that person. He was on this board. Went into what we thought would be a short meeting. 8 hours later, we walked out, and I was the chair of the board. And as the only woman and the least experienced business person in that group, I suspect there were motives there. However, one of the things that I do well is run a meeting. Everybody talk to me. I immediately called Don Jacobs for advice, and he said, don't trust the bankers first, because the bankers make their money only if the sale of the company goes through, so try to get an independent banker. If you can, you won't be able to, but try. And secondly, don't get an M&A lawyer, mergers and acquisitions lawyer, whose fee depends on the outcome of the transaction. Negotiate a fee that's independent of the outcome. So the board could agree on the company. It was Crothers in New York, the best M&A firm in the country. We could agree on the lawyer, Faiza Saeed, who had just done the Time Warner merger. So I called up Faiza on a Saturday, negotiated a breathtaking, even to this day, amount of money for her time and effort. And on Sunday, she was in Cambridge, and we were off. 6 months later, through a lot of turmoil, a lot of calls to people for advice, we sold the company for $1.6 million. Carl Icahn, who had bought part of the company during that process, sued us. We won. And so it was an interesting and informative thing. And the lesson there is sometimes you're ready for a job that you may not think you're ready for. And the key to being successful in such a job is knowing what you don't know and having people that you can call for advice, unbiased, objective advice to help you along the way. After that gig was over, Wally suggested I talk to his son, John Gilbert, who had started a company called Cytonome, with the vision of building a machine that would sort cells for gene therapy. This was in 2002. It was fabulous. He's a microfluidics guy, and so this sounded like a good idea. I said I would join the company for a year to help him out with the people part of the company. We decided jointly that we would flip roles. I became the CEO. John went to his true love, which is serial entrepreneurism. I took over the company, and over a very demanding 3-year period, raised money for the company after talking to 233 VCs and bankers and so forth before I found a partner during the recession. Now, in 2013, I'm pleased to say that our dream machine is in a facility in Japan being used by Sumitomo for the development of some cellular therapies using iPS cells, a machine we developed for our partner, which was a strange partner. We were interested in cellular therapy. He was interested in making cows for milk. And so he wanted to use cell sorting to sort sperm into male and female sperm so that he could make milk cows. And you don't want milk bulls. They are not good except for their genetics. So he was interested in the technology. We make machines for him. They are now all over the world. They're very robust and easy to use. And our newest little toy is a cell sorter that we hope will democratize cell sorting. Thermo Fisher will release it in a soft release this year. It's a sort of a trial balloon. This machine will only identify GFP positive cells, green cells versus anything else. And the second machine, which is close behind in development, will be a regular cell sorter, but anybody can use it. It's push button. Now, as you heard, I sit on boards, I sit on committees, I do good works, or at least what I hope are good works, including things like the March for Science, where as honorary co-chair I did a lot press. I know what Ira Flato looks like. In my remaining time, of which there's not much, I would like to tell you a little bit about what I have learned over the years, and in fact, quite recently, about why it is that we've been so slow to make progress when so many of us in academia and in business believe that it's important to use all of the talent of the country. These are the 3 books which have most of the information that I'm about to give you. They're very important, particularly Thinking Fast and Slow by Daniel Kahneman, a Nobel Prize-winning economist who studied why people make the decisions they make about finances. If you want to be both entertained while you're educated, read Michael Lewis's take on that work. He's the guy that wrote Moneyball. Blind Spot tells you everything you could possibly want to know about implicit bias. It's a very academic book, which means that it can be boring at times, but it is thorough. You can get a lot of that information in Lean In, which is a very readable book by Sheryl Sandberg. And Whistling Vivaldi is must reading for everyone, because Whistling Vivaldi is about the internalization of implicit bias and how it can turn in on ourselves and impede our own progress. And it's a very short book, wonderful read. This is the takeaway lessons that I hope you'll go with. We have known for a very long time, and by we I mean social and scientists and psychologists, that we make errors in our decision making. And now, at this point in time, data from an enormous number of sciences is converging to make this a very strong theory. It's a theory now that approaches scientific consensus on the order of climate change or evolution. And its basis, I believe, as a neuroscientist, and as I do this reading, comes from the fact that our brain, wonderful as it is limited in its ability to deal with the massive influx of data that it gets every nanosecond from the senses. And so it's had to evolve mechanisms to ignore most of that data. Now, the fact that this is the result of evolution doesn't mean it's not changeable. After all, we are masters as humans of making up for our deficiencies, our evolutionary deficiencies. We don't have fur, we invent clothes, we invent housing, we figure out how to use fire. So we are really good at addressing evolutionary challenges. I am here to tell you something you already know, which is that those of us who have been in sciences, physics, biology, chemistry, math, and so forth, hard sciences, have been particularly resistant to this notion that our decisions might be affected by things that we do not control. And besides, there's a little bit of a bias. We think that there is a pyramid with pure math at the top and the behavioral sciences at the bottom. Think about that a little bit. We'll come back to it. That's really the bottom line. So if we go over, you've gotten it. So the mind does an enormous amount automatically, and we do not control it. So when we encounter information, we make associations, and we make decisions based on those associations, and we have not thought about it. And Kahneman poses it as 2 ways of thinking. There's the automatic, fast system, which you call system 1, and a slow, deliberative, energy-consuming, lazy system, and that's system 2. And it's lazy because you want to conserve energy if you're in the savannah or the jungle and there are threats all around you. You need to conserve your energy for survival. We've advanced a little faster. And you might think about it this way. Dr. Spock is system 2, thoughtful, deliberative, and Homer Simpson is running around all the time doing his thing. Unfortunately, Dr. Spock sits there surrounded by Homer Simpson, and most of the time lets Homer do all of the deciding for us. Well, as of late, there have been imaging studies which show that different parts of the brain light up when these different systems are being used. And this is a very super high order kind of a look at the functioning of the brain. Under each of these areas there's enormous complexity of cell type and connections and interconnections. And so it's a crude look. We're up at satellite level. We certainly can't see, we might see the continents, but we can't see the mountain ranges, never mind the roads and individual addresses. I highlight one particular area here because it's just so interesting. This little thing here, the ventral medial prefrontal cortex, lights up in system 1 kinds of thinking. People who have injuries in that part of the brain have no gender bias. I don't know what that means, but they don't. The other time that that part of the brain lights up like gangbusters is when political issues are being discussed, when we have political associations. A lot of hot activity in that area today, huh? I like to make a little analogy here. We're all familiar with optical illusions. You will look at this and you will see those two tables as different sizes. They are not. They are the same size. But we see them as different sizes because we have a two-dimensional visual system that has to deal with a three-dimensional world. And so the brain is wired to interpret certain visual stimuli as three-dimensional when they're two-dimensional. And so we see these tables as the same size. And even if I trace the table and using that little thing in PowerPoint, rotate that trace and slide it over the table next door, you still will see them as different sizes. Well, implicit bias is kind of an associative illusion. We make these associative incorrectly because of the shortcuts, the habits of thinking that we've developed as part of our mechanism for dealing with this impossible data stream. And we make these associations and these habits arise because of the milieu in which we grow up. They are informed by our culture, the American culture. For example, in this culture, we believe, it's a sort of a universal feeling that women are not as good at math as men. And in this country, women indeed don't score as well on standard exams as men do. This is not the case in other countries. It's the case here. So these are these kinds of habits, and you can understand how these habits develop. If you put your mind to really studying something like the violin, when you're learning how to play the violin, you have to pay a lot of attention to where everything, how things work structurally, where your fingers go, how you hold the bow, how you draw the bow across. By the time you have done your time and you've put it in and you've truly learned how to play the violin and know what those little black dots mean on that white sheet of paper, you don't think about it anymore. It's just you and the music. The mechanics of doing the bowing are now behind you. They have become a habit. When you go to and from work, you don't think about it. You leave home and you arrive at work, and who knows what happened in between. You're really thinking about what's going to go on during work or what happened the day before. It's those kinds of habits which are the same kind of mechanisms that underlie the topic of implicit bias. And I think, and this is truly this is truly a hypothesis, that in this country in particular, where we have not dealt directly with the issues of our history, we don't teach it, we don't acknowledge it, and so we have this haze of racism of all kinds, slavery, the treatment of Native Americans, the Orientals who built the railways, the Japanese internment. We haven't dealt with them head on. We haven't acknowledged that there is none among us without fault. We are human. And so we all share this tendency for implicit bias and the wish to be with our own kind. It's not a white man's problem. It's a human problem. And that is the first step in dealing with and changing with and accommodating this evolutionary deficiency which we have. So good people can make mistakes, and we do. I'm here to say, though, that there are indications that we can deal with this in ways that do not require an enormous amount of time. For example, at the American Society for Microbiology, Arturo Casadval noted that there were an awful lot of panels that seemed to be all male. And so he leaned over to Jo Handelsman, who did a really interesting study on the CVs, like I told, on CVs, where she showed that if you have identical CVs, but one is Jennifer and one is John, faculty, young faculty, old faculty, white faculty, people of color, males, females, will all say that John is more qualified, is worth more money, and he's more likely to be offered a job. And so a decision has been made about those two candidates because Jennifer is not John. So he leaned over to Jo and he said, "Jo, do you think this is gender bias?" And Jo said, "I don't know." So in 2013, Arturo, who was the program chair, said to the committees, you've done a good job. We have pretty good representation of women on our panels. But our society is over half women. And if we want our society to thrive, we really have to do a little bit better. So I want you to think about that as you make up the speakers for this year's talk, and particularly panels where you are all male in the selection committee, because there's more tendency to have all male speakers if it's an all male selection committee. So, think about it. And if you see an all male list of speakers, question it. And, let's do right by our members and our society. And so he did that, and started doing that in 2013, and what these data show is that the number of female speakers, even when all the choosers were all men, went up. And by 2015, there were very few all-male speakers, less than 5%. And the number of women presenting represent the number of women in the society. So this is really simple. It doesn't take a lot of time. One of the keys is it's not saying you're a bad person, you only pick men. It is saying our society is a group of people which is of mixed gender, and so in order for our society to thrive, we really need to work together to make sure everybody participates. It is my hope that this works with people of color as well, but we'll see. Another piece of evidence that says that this is possible to deal with is a double-blind experiment that Patricia Devine and Molly Carnes did at the University of Wisconsin, where there are 92 departments. And so she divided these departments in half, matched them by size and approximate field, and then randomly assigned them. So it's a true random, this is a difficult trial to do. It does not quite reach the power that is needed for statistically definitive results, but it is encouraging. Because what she found was, and this was statistically significant, was that after a 2.5-hour workshop, like some of you heard earlier in this meeting, there was a workshop here, that people felt the climate in the departments was reported to be better if they went to the workshop. Now, this is a faculty workshop. That meant that you could not say, you have to go to this workshop. So only about a quarter of the members of each faculty went to this workshop, and it still had an effect. The interesting to me thing is that she followed these departments over time, continues to follow them, and there was a change in hiring patterns. In that more women, more people of color were hired after this one intervention of a 2.5-hour workshop. Again, this doesn't reach statistical significance, and it can't, because 92 departments isn't enough. So she'll continue to follow them, and she will try to get this thing set up in other places, but nevertheless, for me, it says that it is possible to affect our reaction to our implicit biases if we acknowledge them, if we think about them, and if we do little tricks like saying, well, if this were John instead of Jennifer, or if this were Julia instead of Julia, what would I think? How would I evaluate this? It's very difficult for us in our fields that are represented here to do the true thing, which orchestras did. Orchestras used to be all male, because somehow the females were just never quite good enough. But then they went to blind auditions, where you were behind a curtain. And for a while, that had a little bit of effect, but you really saw an effect when people walked to that blind audition on a carpet, so you could no longer hear the click of high heels. When auditions started being done under those conditions, very quickly orchestras became half women. My goodness, they certainly learned a lot in a short period of time. So this is not something that is a result. There is certainly substantial explicit bias left in our society. But amongst the institutions that we deal with, even though it exists there too, there are nevertheless enough people who think we need to do something about it that it really should happen more rapidly than it has happened. And it is my firm belief that this is why, and if we implement some of these and you go pass on this knowledge to your colleagues, that maybe instead of this very slow curve, we can finally hit that inflection point and move up. Thank you for your time. Do I have time for questions? I guess I do. Are there any? I will be around for lunch and thereafter, so you certainly can find me. But I'd be happy to entertain some questions now. Yes. How are you? I'm good.
A wonderful talk and inspirational as always. So here's an interesting question that as our schools begin to develop, you are on faculty, in faculty to some extent, we find that those individuals are charged with being on absolutely every committee because of trying to increase diversity. And of course that detracts from their ability to create their own research programs to develop as faculty. So
Yes.
I hope we're in a transitional period, as you say, and we'll get to a point where that's not a problem. But right now, I do feel it is. How do you, any advice on what we can do to alleviate your impact particularly?
Well, I think one of the things that some institutions have started is mentoring for junior faculty, where they're paired with a faculty member where there's a good match. And the job of that mentor is in part to say to the junior faculty member, because the chair can't, the chair has an agenda. There are things that need to be done, and he or she needs volunteers to do those things. And so chairs will ask, and they're not going to tell you that you can say no, but you can say no. And so it's the job of mentors, or for you to figure it out amongst yourselves, that you can say no. It is perfectly acceptable to say, I would love to do that. I cannot at this time because I need to be sitting in your chair someday. And so we need to teach our young faculty to say no. We also need to teach our majority faculty to be able to fill in for those and to understand that part of the job is to help to diversify the faculties. That's much harder. I appreciate that. But if we don't protect those junior faculty, we will never get where we need to go. I saw another hand somewhere. Stand up. Yes. I think the main thing is to not be afraid to walk off a cliff. I mean, it's true that I took advantage, but I didn't think of it in that way at that time. It was more, if I don't do this, I'm going to die. Or I don't see any other way out of this box. That was very frequently the case. So those decision points are, although in retrospect, they make a nice story. In fact, they're much messier at the time. And so you need to realize that. It's not an easy process anytime you make a decision which takes you away from what you think is the thing you're supposed to do. So the thing you can do now to realize that is to say, wherever you are in your career, we have a tendency as Ph.D. type, science types in particular, but it's true of all highly educated types. We've been trained and we are learned about a certain thing. And very frequently we define ourselves by those boundaries instead of thinking of the skill sets that those fields require. And if you take away the specifics and think about the skill sets, you will find that all of a sudden you have abilities to do other things that you might not have thought of. So that's probably the most useful exercise. The other thing is I have a short attention span. I'm curious, as most people in this room I suspect are. And so the times that I've made changes have been things that have allowed me to indulge that wide range of interests and the ability to think about different things, which is a really important thing, I think, in terms of just, keeping on, keeping on. Any others? I know I'm holding, yes. Well, I think there are two bits to it. One is I certainly had the 3 o'clock in the morning, waking up crying, saying, why the hell am I doing this? I mean, we all go through that. The other thing was I was in the best physical shape of my life at that time because I got up every morning before I went and looked at my plates where there was going to be no colonies. I go run wind sprints up there's a hill at Cold Spring Harbor, a steep hill. So I'd run as fast as I could up the hill, walk down, then do it again. So I was in really good shape. And physical activity does help. So for me, it was physical activity. For you, it might be something else. You really should cultivate another part of your personhood. Reading, music, I don't care what. Develop another interest that can distract you. It will rest your mind, and oftentimes it's during those times when you're not thinking about the problems that solutions come. So it's okay to be discouraged. I mean, you'd be quite unusual if you weren't, with continuous failure. And I wish that during that period of time I had learned, I had realized some of the things that I was gaining. I gained knowledge and connections that were invaluable, and I just didn't know it at the time. And the other thing was I'm incredibly stubborn, so I just would get up and do it again. All right.
Really made it stronger, but my specific question is, how do you know when you can openly admit those things and be authentic? Because you don't want to come across as someone who's coming to for a job interview and you're saying, well, I made all these errors, but I'm great. You don't want to do that. Where's the stage where you can actually do that, and how do you go about that?
Well, I think it varies for everybody. I mean, I have a very strong family, and none of them are in the sciences. So that was one aspect. I have an incredibly supportive spouse. I mean, that Tony guy, we're still married. It's going on 48 years now, and he's been a rock. We've been mutually supportive. We are in related but different fields, so that's been there. And I think that having friends, developing friendships that last, or that you can have these conversations with. At Northwestern, there was a group of women who would get together once a month. I was just in Chicago, saw a few of them. And these are all very accomplished women. But in that group, that was safe space. We could talk about the issues that each of us were facing. And so having groups like that is really important. And you have to start developing those, the earlier the better. To not have anyone. One person can be enough. And as to when you start talking about that, I do not know the answer to that. The only thing I do know is that if you never talk about it, you cannot properly process experience just by turning it around in your own head. You need to say it out loud, preferably to somebody. I don't know, maybe Alexa will serve that purpose. And with that, I'll thank you, and maybe we should all go to lunch.